Hey everyone, Dr. Jack Audie, and I'm just going to whip you through another technique. This one's called an Avidity ELISA. Avidity. And basically what it's measuring is the strength of bonds between two proteins. And we're not talking about covalent bonds or antibonds here. We're talking about hydrogen bonding, hydrophobic interactions. We're talking about those sort of antibody, for example, an antibody antigen binding. Now that's the number one use for it, but you can actually use it for things like IL-1 beta, which is a signaling protein, and the IL-1 receptor, which is a protein receptor. Now these two obviously bond together. And what it does is it measures the relative strength of that bond, right? And it's a bit of an approximate measure, and there are a few issues with this assay, but it's a great starting point as a ballpark figure as what is the stability and strength of this bond. And this is how it works, right? Are we ready to jump into it? Now, just like all the other ELISAs, it starts with a sticky plastic, right? So this is a 96 well plate, and we're looking at one of those wells, and we're looking at those wells from the side, right? And what we're going to do, once again, is we're going to take purified recombinant protein of one of the two proteins that we want to measure the bonding between. So a good example is a spike protein and the antibody that your body produces to bind to the spike protein during SARS-CoV-2. So we pop it into the well, and because the plastic is sticky, the spike proteins stick to the plastic. And this is very similar to all the other ELISAs, right? You put in a protein that you want to bind to that sticky plastic. We then block it, and as per my previous videos, we block it with a non-specific protein that we don't really care about, such as albumin, which is a very common blood protein. So we might get bovine albumin or cow albumin. And what that's going to do is that's going to stick to the plastic. So now the sticky surface is coated in another protein. So now it's not sticky anymore. It's a bit like getting dust on your sellotape. It ruins the stickiness of the sellotape. Okay, so now the plastic isn't sticky. So if something's going to stay in that well, it's got to bind to the spike protein in order to stay in that well. Then what we do is we put in, say, a patient's plasma, or we put in another protein. We put in what we're interested in binding to this. So in the case of looking at the effects of vaccine on producing good antibodies, say we want to know if SARS-CoV-2 vaccine produces good antibodies, we then take a person's plasma from someone who has been vaccinated, which will contain those antibodies, and we put it into the well. And their antibodies will bind to the spike protein. So the spike protein antibodies, so now they're going to bind. Actually, I'm going to put one on each of them for this assay. It's a bit of a poor antibody there. Here we go. Okay, so we've got spike protein in the well, and it's stuck to the sticky plastic. We've blocked the plastic. Now we've put in a human plasma who's been vaccinated, so they have antibodies that bind to this spike protein. Now, we can then do a number of other things, but in a real clear, simple example is we then put in a mouse antibody that specifically binds to IgG, right? For example, so here we're putting in a mouse antibody that binds to human IgG, and we got this by putting human IgG in a mouse, and then that mouse had that immune response producing antibodies for that. And remember that this region of the human antibody here is the variable region, so it's very different between antibodies. And so these antibodies are all designed to spike proteins, but they won't bind to other proteins because this region is different. But this tail region here of the antibody doesn't vary between antibodies. So we can take any human IgG subclass of the same subclass, inject it into a mouse, and then that mouse will produce antibodies, and those antibodies will bind to all the antibodies. So if this was a flu viral protein and this was a flu IgG, anti-IgG human, the antibody would still bind to it. We could use the same mouse antibody. Now, attached to that mouse antibody, again, will be some sort of reporter. So it could be HRP, for example. And you might be thinking, isn't this exactly the same as an antibody ELISA? And it is. So far, this is exactly the same as an antibody ELISA. Now, this reporter tag right here could be HRP, for example. And we know from the previous videos that HRP can convert a colorless substrate into a colorful product. Right, so we're getting a colorless and it's turning colorful product proportional to the amount of antibodies that are in here, right? So it tells us how strong it is, right? And so we would end up with a 96 well plate. This is now a top view, a 96 well plate. So this is unvaccinated person. They would have no antibodies, so they would have no color. And this is a vaccinated plasma. And so that would have a lot of color. And that's because, remember, we've got loads of this happening. Each enzyme can produce loads of colored product substrate. And then we can measure the color difference there by measuring the absorbance of blue light. So that is exactly the same as an antibody ELISA, right? So the question is how do we test the avidity of the antibody? How do we test the strength of this bond here? We want to test the strength. Now let me just write on this just quickly. So that is human plasma antibody. And this is spike protein. That's from SARS-CoV-2. Right, now we want to measure the strength of that bond there. We want to measure the strength of this bond here. So how do we do it? And this is how we do it. It's so cool. It's kind of random, but it's so cool. So we put in, so all proteins, all of these soluble plasma proteins have evolved in an aqueous solution, right? Membrane proteins in a lipid membrane, but soluble cytosolic or plasma proteins have evolved in an aqueous solution. And so their protein structure is stable in an aqueous watery solution, right? And if we change the properties of that watery solution, we change the structure of that protein, right? We can start to compromise the integrity of that protein. And so what we do is we add to it something called a chaotropic substance, also known as a chaotropin, right? That's the noun for it, so a chaotropic substance. And essentially what this is it's a substance that alters the properties of the aqueous solution, right? So that it changes the hydrophilic, hydrophobic interactions, the hydrogen bonding interactions of the protein with the water, and of the protein with itself. It is a chaos agent, an agent of chaos. It breaks down the structures of the antibodies, right? An example, and I don't expect you to remember this, but an example is this right here, which I think is sodium thiocyanate. I think is what it is. This is a classic example of a chaotropin or a chaotropic agent. And basically, it is a peculiar thing. It's very charged. It's a very polar. It has a peculiar structure. There's a triple bond here between that carbon and that nitrogen. And so this changes the way the molecules within the protein and the molecules within the protein in the aqueous solution interact, destabilizing it, right? So how do we use this to test the bonding strength on it, right? Well, if this bond is really strong, right, we're going to need a lot of this chaotropic agent to dissociate this bond, right? If this bond is very weak, if they're barely even touching, right? Say there's only like 1 or 2 little domains in the protein that are barely making this tickle of an interaction here, then we don't need a lot to break it, right? So if the proteins are holding on like this, say the antibody and the protein are holding on like this, it's very easy to break. You only need to slightly change the structure of one of the domains of these proteins and they'll dissociate. If there's a lot of tightly interacting domains in these proteins, more than just one little thing, you need to change the structure of a lot of domains, 1, 2, 3, 4, right? And then you'll get release, right? So it's talking about how many domains are interacting and what's the strength of that interaction between those domains, right? So what do we do, right? So what we do is we've got this sample here. This might be a control. This might be vaccinated person. And then this might be vaccinated person with a low concentration. Let's go picomolar of the chaotropin. Chaotropic. We'll go to chaot. Oh, I missed an O. I'm not a good speller. Oh well. Okay, so, and then we go same person, same plasma, same blood, and we increase the chaotropic, right? Okay, what we've done there is we have we've set up a row in our plate of our 96 well plate, and we've got a control in here. So say this might have no plasma in it, right? So there's no yellow in it. We know that this one, the ELISA, is 100% going to work. That's just a perfectly normal ELISA. But now we've got a little bit of a chaotrope in there, a little bit of this chaotropic agent in there. But actually, the concentration of that chaotrope doesn't do a lot, right? But now we've got a little bit more of that chaotrope. So maybe we get just a little bit less yellow staining in there. And then we've got maybe just a little bit less, and then a lot less, and then none. Almost no color in that at all. Just imagine that color is spread across the whole well. There's no way to do that with one of these markers, right? So what we can do is we can increase the concentration of this chaotropic agent, of this chaotrope, and we can look at how much chaotrope do we need, how much of this chaotic agent do we need to break the bonds between this antibody and the spike protein or whatever it's binding to. Now let's run an imaginary experiment here. So this is our vaccination person, right? This is their profile, right? And we want to know, well, what does that binding affinity mean? Well, perhaps we have another row in another column in our ELISA. But this is a person who's actually had COVID-19 or SARS-CoV-2. COVID. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Okay. And now we put in the same concentrations of the chaotrope, picomolar, in, you probably actually use more than this. It'd be more like micromolar and millimolar, but anyway, this is just an example. Oh, I've missed something, haven't I? No, that looks right. Okay. So what we could do there is we could do that right there. And now we look at the yellow. We look at how well did the ELISA work. Basically, we're looking to break the ELISA with a chaotrope. Okay. I'm going extra on that one. Okay, so if we, let me just shift this up. Okay, now if we compare the color curves there, we can see we can start losing binding affinity between the antibody and the spike protein at 1 picomolar, right? But if we then have a look down here at a person with COVID-19, their antibodies are still bound at 1 picomolar, even at 10, even at 100, and we only start to lose binding between the human antibody and the spike protein at 1,000 picomolar, right? And so if we were to graph this, let me chuck this on a graph here. So this is chaotropic concentration, and then we have absorbance, right? Let's compare. In blue, we've got a vaccinated person. So we might see something like that. And this is someone who had SARS-CoV-2, right? So what we can see, remember, absorbance is essentially how much antibody we've got there, right? And what we can see is that the person with SARS, the person who had SARS-CoV-2, their antibodies bound stronger to the spike protein because it took more chaotropic agent, more chaotrope, to break that bond, right? And so this absorbance represents the orangey color there. And so we can see the absorbance only goes down, which is a sign that the antibodies are breaking, the bond between the antibodies are breaking. The concentration only goes down at very high levels of chaotrope. Whereas a vaccinated person, this is a hypothetical example, by the way. Actually, the data shows that vaccinated people and SARS-CoV-2 people produce antibodies of the same binding strength. So this is just a worst case example. Let's say I made Jack Orty vaccine and it turned out to be a terrible vaccine. This is what it would look like. And so in the blue line, we've got a vaccinated person and we can see that their antibodies are bound to it at the bond between their antibodies and the spike protein broke at a lower concentration of chaotropic concentration. So we would actually put the chaotrope in before we put this mouse antibody in. And what would happen is, let me just draw that actually, that's a good idea. I'm just going to draw a mini one. Okay, we're just going to do a zoom in on a half a weld. This is a zoom in on a half a weld. Here's our spike protein. Let's draw two spike proteins. Here's our human antibody. Right, so we've got two human antibodies in here. And now we've got our chaotrope, right? So this is our chaotrope. So we put in the chaotrope. And the chaotrope is everywhere. And it changes the properties of the protein amino acid interactions but within a protein and the relationship between that protein and the water. It causes chaos, right? Very simple. It causes chaos, prevents structure going on. Now what this is going to do is this at this concentration, the chaotrope is going to break off this. So now one of the antibodies is going to disconnect, maybe two, maybe all of them. It depends on the concentration, right, of the chaotrope. But let's say we lose that antibody, right? So now we've lost one of the antibodies because of the chaotrope. So then when we put the mouse antibody on, here's the mouse antibody coming on, and that mouse antibody has a HRP, right? And that HRP turns a colorless substrate into a colored substrate, right? Because we've lost this antibody because of the chaotrope, we then wash the chaotrope out before we put that mouse antibody on because we don't want to damage the bonds between the mouse antibody and the human antibody. We just are testing this bond strength here, the bond between the human antibody and the spike protein. So in between, we put on the chaotrope, see if we can break some of the human antibodies off. We wash out the chaotrope, then we put in the mouse antibody, and then we measure how much of the human antibodies survived, right? And so then by increasing the concentrations of the chaotrope, we disconnect the human antibody here. So the human antibody then breaks off. And so we lose that signal as we go down. So another way to say it is we're just seeing how easy is it to destroy the assay, right? How easy is it, can we destroy the bond between the human antibody and the spike protein with an agent of chaos, which is kind of cool. So that's how we do those assays. And again, this is a relative assay. We don't get an absolute number for this physical strength of this bond. But what we could do is compare vaccinated versus SARS-CoV-2 patients to double check that the vaccine is producing as good an antibody with as high avidity Avidity describes the bond strength between the antibody and the antigen, so we can test to make sure that the avidity between the vaccine antibodies and the SARS-CoV-2 patient antibodies are just as good, right? So that's a relative measure of strength of the bond. Thanks, guys.